recording.
All right. Hi, Kushal.
Hi. Should we start? Okay.
Yeah, I think you should get going.
Yeah. Hello, everyone. My name is Kushal Dey. I am a postdoc in Altus Prices Lab, and I'll be joining as an assistant member in Memorial Sloan Kettering starting early next year. Today, I'll talk primarily about disease heritability and how functional annotations can be used to inform heritability. So just to give a little bit of a background, typically with genome-wide association studies, we try to identify which genetic variants are associated with a trait or a disease of interest. And over the years, there have been a large number of GWAS studies performed across many traits. Like in the GWAS catalog, there are nearly 4,000 different traits and more than 100,000 genome-wide significant SNPs with a p-value less than 5 × 10⁻⁸. However, there have been some challenges in translating the GWAS discoveries into therapeutics. Some of these challenges stem from the fact that most of these complex traits and diseases are highly polygenic in nature, implicating many genes rather than 1 or 2 genes. Also, more than 90% of the GWAS associated signals have been found in the non-coding regions of the genome, suggesting more of a regulatory effect. And also in cases where the variant is truly causal for a disease, they tend to have low effect sizes. And as a result of that, if you have a moderate sample size of your GWAS, you may actually miss out on some of these variants. You can check out these 2 references for a discussion on this particular topic. So first, I will start with an introduction to heritability. Then I will talk about some of the methods that can be employed in estimating heritability, especially in cases where we have GWAS summary statistics. And then we will talk about how functional data can be integrated to inform about heritability and what have been the past research works in this particular domain. So what do we mean by heritability? So heritability basically refers to the proportion of phenotypic variants that can be attributed to genetic effects. And now the term genetic effects is a pretty broad term, and we need to probably clarify it a little more. So there are now sub-definitions like a narrow sense heritability, where we are primarily focused on the additive genetic effects and this contribution to the variance in the phenotype. We have also a broad sense heritability, where we look at both additive and non-additive genetic effects. And these non-additive effects can include the G by G epistatic interactions or dominant/recessive effects as well. So we are going to primarily focus on the narrow sense heritability, and in fact, a subcategory of narrow sense heritability, which is explained by the genotyped SNPs in a particular assay, the HG square. And this is the parameter that is of particular interest for a lot of these GWAS studies for traits and diseases. One way of formalizing this is that HG square is the maximum proportion of the phenotypic variance that can be explained by a linear combination of the genotyped SNPs. So if you look at the formula here, what we are trying to do is find that optimal linear combination W of the genotypes G, such that this particular linear predictor is most predictive of the phenotype Y. And this heritability or SNP-based heritability depends on multiple factors. It first of all depends on the population that you are studying, also depends on the phenotype that's of interest, and also the set of SNPs that you have genotyped. You can also define a similar quantity called GWAS square, where you restrict yourself to variants that are genome-wide significant variants instead of looking at all the variants in your assay. Now, ideally, what we would expect is that the HGWAS square is going to be pretty close to HG square. And that would be the case if we were able to identify all the causal variants and if we had a lot of sample size for that matter. But that's not always the case, and oftentimes it is not the case, actually. An example of that is in schizophrenia, where the H square GWAS is 0.07 and the HG square is 0.24. So it's almost 3 times lower than the HG square. And this gap is called the hidden heritability. And one way of closing this gap would be to have larger sample sizes and or probably meta-analyze across different cohorts and try to bridge the gap. And here's an interesting paper that just came out probably last week or maybe a couple of weeks and from today, and where the authors actually looked at meta-analyzed genome-wide association studies across 5.4 million individuals, and they're primarily interested in the heritability of height. And what they observed was nearly 12,000 independent loci that were significantly associated. And also the most important thing is that they actually, these 12,000 loci together managed to explain nearly all of the common SNP-based heritability, meaning that we have been able to mostly saturate the heritability signal with all the GWAS associations for height. However, we probably need similar number of individuals or even probably more for some of the other trait or disease-based studies. Discussion of that can be found in the O'Connor et al. 2020 Nature Genetics paper. So now I will talk about some of the methods that can be employed to estimate heritability. And here I will primarily focus on GWAS studies of largely unrelated samples. So we're not going to talk about twin studies or family-based designs. And also, in the interest of time, we are primarily going to talk about one of the methods, the stratified LD score regression method. But it doesn't mean that there is any dearth of other great methods in this particular space. We will, however, start with one of the preliminary models for estimating heritability in relatively unrelated samples, where we have the phenotype vector Y and then the entire genotype matrix. And in such a case, we can actually have a mixed effect model where we have some fixed effects due to some covariates, and then the U here is a random effect for the genetic effects, and the epsilon is a random effect corresponding to the environmental effects. If we think about like a variance component analysis style of nomenclature, we can actually write down the or decompose the variance of YV into two components, the sigma squared G times A for the genetic effect, where A is the genetic relatedness matrix that can be computed from the genotype data, and then sigma squared E times I is the environmental component. The heritability AG square is basically then the fraction of the variance of the phenotype that is explained by the genetic effects. However, and so
sorry, can you clarify what the the covariates that you're mentioning here in X that includes the genotypes, or it's not?
So the covariates can include some of the other like age, sex, or some of the PCs, for example, that you can get from the genotype matrix. It is the main genotype matrix that goes into the A. So A is basically the genotype matrix times the transpose. So that's the genetic relatedness matrix that we can construct from.
I see. Okay. Okay. So the random effects is including the genetic. Okay.
Right. Yeah, thanks for the question. One of the constraints or bottlenecks with using this particular model, however, is that you need to know the full phenotype vector for all the individuals as well as the genotype matrix. And that information you may not always have, because either it's a large data set that you have to kind of share around, and also because of privacy concerns. So one of the decisions that was however, taken as part of the Nature Genetics editorial in 2012, where they actually asked the authors working on these GWAS studies to publish or maintain a database of the GWAS association z-scores and p-values for all the variants that they have investigated, all the typed and imputed variants, and not just the GWAS significant hits. And also to maintain a database of the sample sizes based on which these z-scores were computed. These GWAS summary statistics are kind of widespread now for most of the traits. We do have GWAS summary statistics from different, either from different biobanks or from other independent studies. One of the constraints, however, is that we do not always have the LD computed from the individuals that went into these studies. And a stopgap solution there is to use an external LD reference panel, because we need to understand how the SNPs are kind of correlated with each other, where we know that proximal SNPs tend to be highly correlated because of LD architecture. So one option there is to use external LD reference panels from other consortia, like UK Biobank is there. There is also 1,000 genomes. And the question then was that if we are given a GWAS summary statistics and maybe an external LD reference panel, can we still go ahead and estimate the heritability? And this is where the LD score regression method kind of comes into play. The LD score regression mainly was built around the intuition that SNPs that are in higher LD with other SNPs tend to have a larger test statistics, like a higher magnitude of z-scores or higher chi-square statistics on average for a polygenic trait. And this is purely because of the fact that if you have a lot of SNPs that are tagging the focal SNP, and for a polygenic trait, we know that there are causal variants kind of distributed all across the genome. There is also a possibility that some of these tagging SNPs can include causal variants for the actual disease or trait. And that can elevate the chi-square statistics for the SNP that you are interested in. One way to kind of formalize this is by defining what they call as LD score for a particular SNP. So the LD score is the sum of the squared LD between the SNP X and all the other SNPs. And what they observed for schizophrenia was that if you plot the mean chi-square statistics for different LD score bins, then there is a positive association between the two, which basically matches up pretty well with the intuition that the authors had in the first place. What was, however, quite interesting is that now if you actually build a model to regress the chi-square statistics on the LD scores, if you look at the slope of that particular regression model, and this is based on the mathematical formulation, the slope is actually a function of the h squared g, or the heritability. And in fact, the slope is h squared g times n by m, where n is the number of samples and m is the number of SNPs that you are looking at. And both of those information are known. So if you can actually compute the S, you can also get an estimate of heritability. There is also another factor here, which is the intercept I. So the intercept I, mainly if it is not equal to 1, it captures more of the confounding effects like population stratification and all that. The next question, however, is that, okay, we can actually compute the heritability for a particular disease or trait based on all the SNPs that have been genotyped. But what about some particular category or functional category of SNPs? Like, is it really true that the heritability of a particular category of SNPs, let's say a conserved SNPs, or maybe SNPs that are in DNAs elements, they have higher heritability contribution compared to the SNPs genome-wide? And one way to frame this is by looking at the LD scores in a way where it is like, previously the LD score was just a function of a particular SNP, but now we can define LD score for a SNP and a functional category F in such a way where previously when we were looking at the sum of the R square, we were doing it over all the variants, but now we can constrain ourselves to only the variants that fall in those functional categories. And then once we define this LD score X, F, we can actually take these scores and use these as predictors for the chi-square test statistics, to predict the chi-square test statistics. And the tau F here is the contribution for each of these different functional category based LD scores. And the heritability Hg square F can be written as a linear combination of these different taus. And if you have an estimate of Hg square F, you can basically compare it. You can look at the per SNP heritability in that category, which is Hg square F by MF, where MF is the number of SNPs that are in the functional category F. And you can compare that with the genome-wide per SNP heritability, which is Hg square by M. So that will then give you the overall estimate of the heritability enrichment for that particular category. What's also interesting is that in this particular model, as you can see, that this is a multiple linear regression model with multiple different LD scores for different functional categories, all kind of used jointly here. So the tau F is kind of the conditional information that we are getting from one of the functional categories, conditional on other functional categories that you can have in the same model. In 2017, Stephen Ghazal from Altes Prices Group, he extended this particular framework to also use continuous annotations. The overall mathematical formulation is pretty similar to how it was for discrete functional category of SNPs. However, he also introduced this metric called tau star, which is actually a scaled version of the tau, and it accounts for the standard deviation of the annotation, which is also a function of the size of the annotation. And the tau star is primarily defined as a standardized FX size of a particular annotation queue, conditional on the other annotations in the model. So going forward, I mean, we will talk a lot about these two metrics, the heritability enrichment metric and the tau star metric, as those are the two metrics that are typically often used in terms of understanding whether a functional category or a functional continuous annotation is important for a disease or trait. Although here we are primarily focusing on the LD score regression, but there are also some other very cool methods in this space that I would like to point to as well. So there is the LD eigenvalue regression. So one of the constraints with the LD score is that it only looks at the sum of the R square, which is basically the diagonals of the squared LD matrix. And the LDER kind of generalized that by looking at eigen decomposition of the squared LD matrix instead. So this is a method from Hongyu Zhao's group. There is also the Hess method, when Boshi led this particular work in terms of estimating and visualizing local SNP heritability. From Doug Speed and David Baldwin's group, it relaxes the polygenic assumption that goes into the LDSC model, and also allows the users to flexibly specify a heritability model. There's also a discussion of the comparison between the Samher and the LDSC method presented in the Ghazal et al. 2019 Nature Genetics paper. Another approach where you can actually use heritability to prioritize genes for a disease, it was performed by Omer Weissbrod's group, and that's the benchmarker paper.
So can I ask,
yes.
Amongst these methods, which is optimal for what situation, and why is LD score regression the most popular?
Yeah, so I think this LD eigenvalue regression, it just came out this year. So I think more formal comparison is still left to be done. One of the things they, however, show is that when it comes to estimating the standard errors of the heritability, their standard errors are often much lower compared to that of the LDSC. Now, for the Samher method, I mean, this method had a different, they had a different recommended model to the LD score regression, but in the Stephen Ghazal's 2019 paper, what he showed was that the LDSC actually is, or stratified LD score regression, basically using all the functional annotations, is actually performs better than using Samher with the particular annotations categories that they chose. So right now, I think when it comes to the overall analysis of the different functional annotations and the heritability enrichments for those annotations, and whether that matches up with that of the fine map SNPs, the LDSC seems to be still the best candidate to use, but I think there's definitely some scope for further comparison and benchmarking of LDSC with some of these other methods like the LDER.
Can I ask one other quick question? If you're running out of time, tell me to keep quiet, okay? But
no, please go ahead.
I really like the continuous formulation, but I'm confused why people usually use a discrete formulation, right? So for example, you take DNase peaks, attack peaks, histone marks. The quantitative signal is a very, very important unit because there's a lot of noise in the peak calling, right? So yes. Why does everybody use discrete annotation? Is it because that's the easy thing to be provided, or is there a reason does a discrete version work better, or why not use a continuous version always?
Yeah, I mean, you can actually use a continuous annotation. Oftentimes, what happens is that if it is just the continuous values are kind of more noisy in general, so that can somehow bias the signal to some extent. And whereas in those cases, maybe looking at a discrete or discretizing that continuous annotation into different categories often can give you a better signal in that sense. But if you have a well-powered continuous annotation, which is kind of meaningful, it can often be used as well. And it has been done for several annotations where we have used continuous annotations. One other thing is that oftentimes if we just go by the continuous annotation, so typically there is a difference between a probabilistic annotation and the continuous annotation. So if it is a probabilistic annotation, then it kind of makes life a little easier to compute the enrichments and all of that compared to if it is just a continuous annotation. And let's say that if we have a probabilistic annotation, then also there is often an issue of the fact that if you have very small probabilities overall, then the overall annotation size can be quite small. And when the annotation size is small, then the tau stars or the tau values kind of tend to be a little noisy and less well powered. And so in such cases, it helps to discretize at a level where we have a particular annotation size to use ideally.
Awesome, thanks.
Thanks. So now we are going to talk about how functional data can be used inside the LD score regression of the stratified LD score regression framework, and how it has been used so far to inform about heritability. One of the papers, so this is from the Finucane 2015 paper, the original certified LD score regression paper, where she looked at different functional regions from coding, enhancer, super enhancer, conserved regions. And what was kind of found was that a lot of these regions are enriched for heritability for multiple traits. And at the same time, there are some traits for which some specific functional regions are more important than the others. So for example, here you can see that for BMI, she observed a very high enrichment in conserved regions, but that was not always the case for the other functional regions like H3K4 and E3 for that matter. One other question was that this enrichment of heritability in the functional categories that we are seeing, why are we seeing it? Are we seeing it because the causal variants in these categories tend to have larger causal effect sizes in general for GWAS, or do we see more number of causal variants from GWAS in this functional category? So this was actually resolved by a paper from Luke O'Connor in 2019, where he looked at polygenicity enrichment against the heritability enrichment and observed that a lot of there was a close correspondence between the two, kind of highlighting that this functional heritability enrichment is probably more due to having more number of causal variants in these functional categories than like higher causal effect sizes in general. There was also another analysis done by Farhad Hormozdieri, which was looking at the GTEx EQTL data in whole blood and meta-analyzed across different tissues, and analyzing the heritability enrichment and the tau star values for the different traits. And these were all meta-analyzed across a lot of traits. What he observed was that there was definitely some enrichment for all CCQTLs in general, but he had this max CPP score, which was basically based on fine mapping. And when he looked at this fine mapped CCQTLs using this max CPP score, he observed much higher enrichment and also much higher tau star, both for whole blood and for meta-analysis across multiple tissues. One other direction that was kind of taken by Hilary Finucane in his 2018 paper was to look at the genes that are specifically expressed or enriched in expression in a particular GTEx tissue compared to the other tissues. And she called these genes as the SEG genes, or specifically expressed genes. And she annotated or generated a functional annotation based on all the variants in a 100 kb window around these SEG genes. And she then analyzed those functional annotations for different traits. And what she found was a high specificity of some of these traits for related tissue SEG genes. Like for schizophrenia, so this is from both the GTEx data and another data set that she looked at, where the CNS cell type and tissue-based SEG genes were very, very highly significant for the schizophrenia GWAS heritability enrichment, but that same thing was not the case for the other tissues. And the same thing was true for rheumatoid arthritis, where she observed signal for blood or immune, but not for many of the other tissues. And a similar analysis was recently performed by Briars et al. 2020, where they looked at different brain gene programs and analyzed both using another gene score magma, which is not heritability based, and the LDSC, which is heritability based. In most of the cases, they observed a close correspondence in terms of the enrichment results between the magma and LDSC. And also, as you can see, that they observed that for a lot of these brain traits like schizophrenia, intelligence, Parkinson's, or even MDD, the brain-related genes are much more important in terms of heritability enrichment compared to some of the other tissues. One question, however, that still remained was that, okay, we can actually identify what are the possible critical tissues for a particular trait, but can we actually identify the cell types that are actually important for these traits? And can we try to distinguish between which cell types are more prioritized for a disease compared to the others? And this was an effort that we kind of led, where we looked at single cell RNA-seq data from a large number of tissues in the human body and looked at gene programs that are defined by genes that are specifically enriched in expression in a particular cell type. And we linked those programs to variants, not using the 100 KB window as was used in the SEC paper, but using the activity by contact and roadmap and answer gene links. What we observed was that we were able to pick out the right cell types for the right blood cell traits. As you can see here with the B cells and T cells being more enriched for lymphocyte count, erythroid more enriched for RBC count, etc. And then we can actually extend this to an analysis of all the immune-related diseases, where we see that B cells, T cells, dendritic cells have a lot more signal for different immune-related diseases compared to something like erythroid, which doesn't have that much of a signal. Finally, I would just like to flag two other works in the single cell space linking to GWAS. One, this SCDRS method, where the authors looked at scoring cells in an RNA-seq data based on the GWAS information and the gene expression information at the level of a single cell. And the similar kind of effort was pursued by Fulong Yu from Vijay Sankaran's group, where he looked at ataxic data and scored cells based on combining the ataxic information at a cell level with that of the GWAS. So that's most of the material that I wanted to present. And I would like to thank all the authors of the different papers I presented and also the researchers that I mentioned who are represented today.
Yeah, the last three methods are really interesting, and I'm curious whether you can sort of try to give some insight into what's the relative advantage, disadvantage. I mean, obviously one is attack versus RNA. Has anybody looked at correspondence, like with multi-ohm data? Does scavenge and SCDRS tell the same story? SC-linker and SCDRS. I guess SC-linker has a better like gene model, is that right, compared to SCDRS, the gene selection model?
Yeah. So one difference between the SCDRS and the scavenge is that, so scavenge is a method that actually looks at fine mapped GWAS SNPs and then combines the fine mapped GWAS SNPs with the ataxic peaks, whether we see ataxic peaks in that particular cell overlapping the fine mapped GWAS SNPs in general. And in that paper, they primarily focused a lot on the different blood cell traits. And one of the constraints is that the fine mapping often works very well for some of these well-powered quantitative traits compared to some of the disease traits for that matter. And so, so I think in that sense, the SCDRS is probably more expansive in its use because it basically uses the magma gene score and then links that with which genes are more expressed in that particular cell. And the magma gene score is more stable and you can generate that for many types of GWAS data sets and pretty well powered at that. Regarding the SC-linker method, I think one of the things that mainly drives the signal, and we do show that the SC-linker outperforms magma in terms of the sensitivity and specificity for the right cell types and the right traits. And we believe that much of that actually comes from the fact that we use this enhancer gene links in general, which is something that was not used in the SCDRS framework. So in that sense, I mean, and also like, as you mentioned, that the gene programs at the level of cell type are kind of well powered in that sense. We kind of aggregate information across multiple cells to kind of define these gene programs that way. So yeah, I mean, SCDRS, I mean, one of the uses of that could be for visualization and visualizing what could be the cell states and maybe figure out like a region that is of potential interest. And maybe then one can also look at the genes that are primarily expressed in those regions and then look for the heritability analysis as well. That's the possibility.
Great, thanks.
Thanks, everyone. I think I overshot a lot of time. Is Gao here?
Yeah, I'm here, Kushal.
Okay. Okay, so Gao will talk about fine mapping. So Gao is an assistant professor of neurological sciences in Columbia University, Department of Neurology, and he previously did his postdoc with Matthew Stephens. And I think there are two methods that are really popular and many people have probably used. One is the MASH work and the other is the SUSY work. And I think Gao will probably talk more about SUSY or maybe even MASH, I don't know.
Thanks, Kushal, for the kind introduction. Well, Well, unfortunately, I'm not going to talk Susie this time. So I guess, sorry, the mesh this time, just focusing on Susie for now. Well, I think, yes. Okay, maybe that's a good point to start. So we're going to discuss about file mapping today. I'm going to focus on motivation methods and some practical considerations. What I'm not going to focus on is some recent extensions that we made to the file mapping framework, that including combining the multivariate analysis of mesh to file mapping multiple traits, and also methods for file mapping different molecular phenotypes, such as the QTEQTLs, splicing QTLs, especially methylation QTL, where you can treat things as a functional data. But those are more like derivatives of the basic file mapping framework. And as applications, for sure, we can also, what I'm not going to talk today is using annotations in the context of file mapping, because Omer is going to talk about right after me. So we're going to focus on something like really basic concepts and some practical considerations. Let me just start by some kind of routine introduction of where we are in this picture. Of course, panel A here is what we do in GWAS, right, on this side. And traditionally, we talk about the statistical genetics. We're actually focused on the mapping for disease gene directly from the genotype data, including linkage and later association studies. But in this context, sometimes people refer to that as functional genomics. It usually was like RNA-seq computational biology. I was a little surprised when they say, oh yeah, I don't do genetics, I do RNA-seq. Some people describe themselves that way. But as you can see right now, things are kind of merging together. As Kushal has shown in his excellent lecture already, that got the SNP to the genetic to the gene level mapping, and then you can identify the gene level interesting genes and networks in a context of the functional genomics, and of course the genetic component of the functional genomics, right? Because there's other contributors to it, just like any complex trait. But on the other side, on the B panel is where we are interested. We're going to find the actual, what we call the putative causal variant, and that seems to have some functions in it, right? So the trick there are like, the first thing is to control for the correlation between the variants, as Kushal was introducing already in his lecture, due to the LD, and but itself in the context of LD score regression, you sort of want to try to average and aggregate all the effects, all the small effects together, considering LD. In the case of file mapping, you've got to distinguish, if you can, between the putative causal and the non-causal ones given the LD information. And the other important aspect is, of course, with the putative causal variant, what functional annotational features that they have in the genomic sequence context. And that will be related to Omer's lecture later. So today I'm going to focus more like on identifying the putative causal variant in the presence of LD, and what's the method and tricks going on there. In terms of applications, this is very good to find causal variants and understand biological mechanisms. Although you could imagine not really looking for the mapped causal variant, but just aggregate evidence together at the gene level, you can still discover important gene and pathway as therapeutical targets. So these two efforts are both important and should be pursued in parallel. So a very basic illustration of what this in terms of file mapping in the context of a linkage disequilibrium is that when you have a marginal summary statistic from your GWAS study, you probably get the effect size estimate on the y-axis, and then you look at this, these are like both of the two SNPs seems to have an effect. Although you know that, well, suppose you know that the right one is the causal SNP, but there's a non-zero effects in the other one, and they could be highly correlated. So the goal here is to color this red one from the green one, not to consider their combined effects, right? So in practice, we may see Manhattan plot like this, where you see people used to just color, the GWAS hits by Manhattan plot in different chromosomes. But if you zoom into one region, what people typically do in terms of visualization is to pick the top signal, or they call it index SNP, defined by the smallest p-value in the region, and then to look into also other SNPs in different levels of correlation or LD between them. So here the definition for LD is simply the Pearson's correlation R squared. So in this case, as you can see, even with this index SNP, it can pick up a bunch of very highly correlated SNP, and there are obviously something else of interest, but then which exact one to follow is difficult, and especially when many SNPs are in LD with each other. So are you going to still pick one SNP, or you want to consider all of them, or at least a few of them? So this is what the file mapping in practice become a trickier question when you have complex situations like this. And I want to also clarify that, in the context of the genetic architecture, what file mapping is designed for is to capture these sparse effects and not the polygenic background. I mean, there's no contradiction between, polygenic versus, non-polygenic type of disease architecture, because, usually in practice, it's a mixture of the sparse and polygenic effects, right? So for example, in this case, it's a schizophrenia Manhattan plot. So you can imagine, maybe, well, just for the sake of argument, maybe we'll ignore everything, not file mappable is what we define. Maybe, below 10 to the minus 6, we know there's some signal going on there. We're not designed, the method is not designed to capture those signal. It's probably like, you have some distant signal already. It's just too many things in LD with it. But instead of only picking the index SNP, we look into all of them and try to figure out, from all these candidates, which ones are the putative causal variants. Of course, traditionally what people do is sort of like a conditional regression approach. The argument is very simple. You know that there are some causal variants in there. You also know many others are in LD with it. So maybe let's start from the most likely one, which naturally will be the index SNP, right? You assume there's one signal there, you pick it, and you regress out the effects from the rest of your signals, and you see if another one pops up. Because when you see another one pop up, then you know that it's sort of a independent signal, because otherwise that will be regressed out. So this is what people usually do, is to do conditional regression starting on the lead SNP. But what's the problem with this approach, right? I mean, first of all, this is not exactly a model; it's an algorithm. It's more like a simple variable selection algorithm, and but that's a technical consideration. The most important issue there is that sometimes there are situations that can be complicated, and this model fails. This method fails. For example, this is the toy data that we created just to illustrate the challenge. As this is like a region of on a genome with a bunch of variants here. You can see it's roughly 1,000 variants. But then this is a Manhattan plot from association studies. So say we zoom into this smaller region, we want to pick up, what are the causal variants from this 100 kb region. And if you only look at this picture, you'd imagine, okay, so maybe there's something going on here where the p-value is close to -20. So let's pick that index SNP and regress it out. So we're kind of removing all the signal in between 600 to 1,000, these ranges. And then you end up having another, seems to be an independent peak, right? So maybe then you take the second best over here, and then you remove all the signal here. So maybe the money will be on, okay, maybe it's something here, and then something here on the top, right? But what in reality is, none of the two so-called causal SNPs are the visually top SNPs on this Manhattan plot. And why do we know that? Because it's simulation data. We can do it. So the effect size we know, but the LD structure is from the real data. So it could happen, obviously, in the real world that two SNPs that are not the top SNP can be a causal SNP. And if you go from top SNPs, that's not going to work. So what we are trying to do then is, really trying to find essentially all these combinations of the SNPs that seems to be explaining most of the signals, right? All in terms of, the largest likelihood, while still keeping it kind of sparse. So this is sort of, the intuition is, if you really assess the likelihoods more formally, comparing the likelihood from, assuming the lead SNP, the index SNP is the causal SNP, and the others are like, non-causal, compared to just looking at pairs of the actual causal SNPs, the likelihood in this case should be higher. And then the type of inference that we actually do is to use the Bayesian variable selection for fine mapping, because this is given the input data like this, it will consider all this evidence contributed by many SNPs and somehow aggregate those evidences, which I'll show in the next slide. But the important quantity here is you can aggregate all the evidence contributed by different combination of models into what's called the posterior inclusion probability. So this is a probability. It also has interpretation of 1 minus the local false discovery rate. So this probability corresponds to a discovery rate, and this is automatically you don't have to do multiple testing correction per se. And in this case, you may end up having a situation where those are highly correlated up to 90% of a correlation. A posterior inclusion probability will be clear enough that this one is much more likely to be a causal variant than the other one. And another important quantity that you want to measure is what if sometimes you're if the LD between these two variants are exactly the same, then do you just pick one out of it, or you want to consider the possibility that both of those are equally likely to be the causal variant? So that's another feature of the Bayesian variable selection that I gave you. So in this case, you have to say that they have equal probabilities, 50% each, and you can't distinguish between them. Although you can't distinguish between them, at least you can sort of cluster them. For example, okay, so sort of make a statement of given a bunch of variants that I can't distinguish between each other, then what's the probability of this bunch of variants containing 1 independent signal, 2 independent signals, and so on and so forth. So there are different ways to formulate this formally as credible set. And the type of credible set that we think is the most intuitive is what I said earlier, is you can find a credible set, for example, 95% credible set, interpreted as the 95% probability or up that this set contains exactly one putative causal variant. So this is can be visualized as you got a causal signal, putative causal signal there, but then you have it exists with a bunch of the others, but that they form into these signal clusters. And the formal definition of this could be found in our paper. I think since then some other tools are also starting to adopt this definition. But before that, people usually refer to 95% credible set as something quite different. So this is a review of up to 2018 what people do for the fine mapping using the Bayesian variable selection approaches. And so as you can see, there are many methods developed, and there are many practical considerations of these methods. So the challenge, all these methods are using the same, or let's say very similar models. It's just the way they solve the models are different, because it's especially in the search algorithm, and they have different ways to come up with the search algorithm. But why is such a big deal? Because the inference kind of done for the Bayesian variable selection model was what I explained before. Ideally, what you have to do is you have to consider the scenario of all the combinations of variables. We already established you can't start from the top one. There's no easy way, you just can't take that shortcut. So if you do evaluate all these configurations, what we call the combination of variable as configurations, you essentially have to do, starting from assuming one causal variant to all the way, all of them are causals, you have to got 2 to the power of p, where p is the number of variables you consider in a space. And for each of the configuration, you compute the probability or base factor in this case, and that the configuration is the causal configuration, and then you can average that as a posterior probability. So that will give you the posterior probability of each of the configuration. And by aggregating different configurations, for example, here, for the first and second configuration, probability is up to 25% each. But for the rest of them, there's nothing there. So for the first SNP, aggregating these two, the posterior inclusion probability is 0.5. So on and so forth. That's how you formally make this inference. But as you can understand, enumerating all these configurations and compute the base factors, that's a major challenge, especially when P is large, right? T to the P is an exponential problem. So that's the number one challenge. And the second challenge is, how do you compute the credible set, or do you ever compute credible set based on these, posterior inclusion probabilities? Because here, the notion of a credible set doesn't come naturally. You have to do something ad hoc in order to compute. And there's no notion of, what configuration capture exactly one causal variant, because, those are just enumerations. And when you aggregate, you don't really, distinguish between these different scenarios. So to give an example of, the challenge of quantifying uncertainty in variable selections, if you think of the true causal effect as, like the first, you got 4 SNPs here, 1, 2, 3, 4, right? And your true effect should be, let's say, SNP number 1 and SNP number 4. But then your SNP 1 and 2 are in LD with each other. Your SNP 3 and 4 are in LD with each other as well. So then if you don't do any Bayesian variable selection, you don't quantify any uncertainty, you're going to randomly pick one out of 1 and 2, which by chance you got that right. I mean, with Lasso, right? If you pick another one randomly from 3 and 4, then somehow, it's wrong. It's how Lasso was implemented. But if you use the conventional Bayesian sparse regression by going through the process of computing the configuration, aggregating the posterior inclusion probabilities, you get that correctly. You correctly assign, those as equal probabilities, 0.5, 0.5. But then in terms of at the credible set level, all you can say is that, we know there are 4 variants proposed here, and we know that out of the 4 variants, 2 of them should be causal, because when you aggregate the peeps, 0.5 plus 0.5 plus 0.5 plus 0.5 is 2. You expect to see 2 causal variants out of the 4. So that's not as ideal as if you can make a statement, say, I identify actually in this case, the same peep, but they should be falling into 2 independent clusters. One of them should have exactly 1 causal variance, right? So this is the methods, the sort of the motivation behind SUSY was these 2 motivations. First of all, can we get this posterior inclusion probability very fast, right, efficiently? And secondly is, can we really, fine map to the single effect resolution where you can make a statement like this, not traditionally the other way around. So here I'm just going to give a very high-level description of SUSY, is that the model itself is written a little bit differently. So here, the first line y equals x to b plus e, that's a conventional fun mapping model. If you depending on the prior for b, right? But that's like a conventional regression model. But in SUSY, we sort of try to decompose b with a bunch of so-called single effects, just like this. Suppose there are 3 effects in B, but in this case, well, of course, we don't know the truth, we have to infer this. But at least we allow for a construction to make this inference. And then we fit the model, instead of we fit the effect with the, on the left-hand side, we fit this whole sum of single effects on the right-hand side. And the theoretical justification behind this setup is, this is approximation, variational approximation to as if you evaluate the entire model space. And that's what this thing on the left-hand side refers to, all the possible combinations of configuration that I showed previously. Or here you can approximate that with a specific structure. And in our paper, we sort of show why this approximation works and how exactly it works by implementing a new algorithm specific designed for this kind of regression. And it works very efficiently, that's the bottom line. So if we go back to a real world example illustration, using, well, of course, real world genotype to illustrate the complexity of the LD patterns. But for this scenario, the inference ends up actually being like this, where we do capture 2 credible sets, right? And each of the credible set contains that causal variant. But the first one here, it refers to the one over here. And this one, as you can see, even though the marginal p-value is not that large, but the posterior inclusion probability here is high, because not many SNPs are in high LD with it. So we can do a pretty good job distinguishing it from the others. Although visually it's not very clear, that was the case. But in fact, so the algorithm naturally gives you that solution. For the other one, it's trickier. Actually, in this one, we got 37 SNPs in this set. And because it's meant to contain 1 signal, so the largest peak, of course, here could be 1. So on the ballpark, 1 divided by 37, you can imagine for each SNP on average, the peak is going to be very small. This is actually another practical consideration, is when you look at the high inclusion probability here, and you want to do, say, functional validations to this particular SNP, sometimes the decision is made not because this SNP has such a strong effect, but just because it's mappable. Because otherwise, you've got to try, that the effect here may be stronger, you've got to try 37 things. Maybe in this case, if you still go by PIP, yeah, relatively speaking, maybe you pick this up, after a few trials. But still, the idea is that there's a distinction between mappable versus having a strong effect. And that's actually by chance, lands up in a tricky region with the LD is difficult. And that's why, using annotations on top of it could actually help with this situation. So the way this works, we do actually have a cartoon, but it just doesn't work on this browser. So I'm just going to say quickly, if you start with what we call the strongest marginal association, our algorithm, if you iterate only once, it's still going to pick that up, although much lower. But after a few iterations, the top one will be dropped. The slides are available on the Google Drive. So if you actually click with Adobe Illustrator, Adobe Reader, you can click on it and you see how it iterates, how the fits iterates. And that's also a feature in our software that we can actually make this kind of plot. I don't think many people do that, but it's just sometimes as you wonder about how the model converges to your solution, then you can actually make this plot from our software and check for yourself. Okay, so I'm going to use the rest of the time to discuss some practical considerations, especially using summary statistic, which as I think most people do with our with this file mapping application anyways. So we have to first of all, define what summary statistic is. Of course, I think many people might be familiar with, what z-scores are from univariate associations. So it's kind of approximately equal to the effect size divided by standard error, where effect size here follows like a linear regression. So if you use the ordinary linear least square estimate, so this will be where you compute the effect size and the standard error for it and the z-score. So there's a distinction between sufficient statistic versus summary statistic. Usually, I think people mostly use the z-scores and the r-squared, the genotypic correlations for summary statistic. Although the sufficient statistic is going to give you exactly the same results as the original data. There are advantages using the sufficient statistic versus the summary statistic. Even though you have the original data, because computationally, if your sample size is very large, much larger than the number of SNPs, then using the summary statistics can give you additional computational advantages. There are two types of summary statistics-based models. The z-score based model, actually, the advantage is you can use the z-score and the r-score, the LD matrix is good enough. You have to make some assumptions in terms of especially the heritability. So when you assume heritability of any single SNP is small, then it's actually pretty decent for GWAS settings. File mapping. That's why many of the earlier file mapping methods use z-score for GWAS, it's fine. But if you use the summary statistic for QTL mapping, sometimes the effect size is very large for like eQTL, then this is not going to be the ideal situation. And you can imagine having the same model, but using the beta and standard error of beta, which we call the BNS model. So of course, these two models have some subtlety in the assumption for the effect sizes as the prior. Although this effect size could be automatically estimated. So that's something once formulated into the SUSY framework can be taken care of. But like I said, important distinction is when you have different sample sizes, and especially when your effect is very strong, then it's better to use the BNS model. And here are some examples of existing methods that use these models. Of course, for the second one, you need more information, but it's better to have it whenever available. Sorry, I'm out of time now, but let me just flash over the other challenge. It's about using the in-sample LD versus external LD. I recommend taking a look at our paper that we have an evaluation for with the current methods. The bottom line is that when you're using the in-sample LD computed from your genotype, then all the methods behave pretty much similarly. But when you have something unusual going on, such as if you use an external LD reference panel, you may have the allele flipping issue. Means that your summary statistic and your LD reference don't even agree with each other on the genomic, on the coding of the SNPs. And that will result in conflict. In that case, something weird will happen. And I think there are many approaches nowadays that, well, not many, but some approaches nowadays to detect the differences for this potential inconsistency. We do also provide another method in our package to do that. So that's one thing to look out for. But even if the alleles are consistent between the summary statistic and the LD reference panels, there could still be, sorry, this is the ways to kind of manage the allele flipping when you merge the data together. But even if the alleles are fine, just because the reference panel comes from a different population compared to the summary statistic, things could still be tricky. But so in our paper, we sort of evaluate also different scenarios and how different methods respond to these inconsistencies. the bottom line is that we still find that our implementation in SUSE so far seems to be the most robust against this problem, and closely followed by the FAMAT program. And the other couple of programs we look into have a stronger problem. But as you can see, if your sample inconsistency is so high, like this, then it's going to be way off the truth anyways, regardless. Maybe some methods behave worse than other methods. For example, here you see that SUSE is slightly better than that, but still very bad. So it's important to actually make sure that, to the best of your knowledge, things matched with each other. This refers to an older results where in practice how things could be matched up if you use a small reference panel versus, this is using the in-sample LD reference, and you got 3 signals here, versus you don't do that, you use some smaller reference panel, you end up having 2 and miss 1. Okay, I'm going to stop here. This is a discussion in the context of a linear mix model, where it says, for the covariate, you have to adjust that of your genotype as well. And the authors show some simulations here. My take is that for linear mix model, this is quite important. I think for the population-based case control data, this is sort of a secondary consideration compared to matching the LD reference with a summary statistic. Sorry, 1 minute over time.
No problem. Any questions for Gao?
Go ahead. Sorry. No, I asked a lot, so you can go ahead.
This was a fantastic presentation. Thank you. Do you have recommendations for, like, say, for QTL meta-analysis, where you're trying to you have summary statistics from different populations, maybe different LD, and what considerations there? Right.
So, well, what we do actually, if they're so different from each other, like 2 different populations, then that's when the MV SUSE, the multivariate version of the SUSE, comes into play. You can have as many populational study groups, and you do that in that setting. But what we do in practice is we actually, sort of what I do, at least in practice, is I evaluate the pi1 coefficient for different studies and see, if the studies are similar enough, or for example, they're from the same tissue, let's say, and then the signals are pretty much close to each other. I'm going to meta-analyze them and combine reference panels. And if they seems to be very different, I still treat them like a multivariate framework. So that's of course, when you only have the summary statistic, that's so much you can do. Fortunately, for most of our applications, we have access to the full data. And in the functional, like QTL mapping, the genotype matrix is not that large. It's usually 1,000 samples is good enough. So we mostly in practice still work with the full data for it.
I had a quick question. So in the Bayesian selection framework, how much of an effect does the prior have? And are the assumptions made? Because I don't know the exact details, but I've heard that some methods like make a pretty strong assumption that there's one causal variant per locus. Now, how much can those assumptions be relaxed and still make the problem tractable? For example, does SUSE also make similar like restrictive assumptions? And if you have functional annotations, can you relax those assumptions? That's a lot of questions.
Good. So SUSE doesn't make the assumption for how many causal variants there. Like FAMAT also kind of relax that, thanks for the improved computation side. But you're right, there are 2 types of priors. I'm going to defer the prior on the functional annotation to the next lecture. So one prior, of course, is on the prior inclusion probability that refers to like functional annotation, given where the variant is, how likely they are to be functional. There's another prior, like given they are already a causal variant, what's their effect size? Like got a specified prior for the effect size. For SUSE, we actually take empirical based approach where we estimate the effect size prior. So it's more or less objective than specifying any random numbers. Of course, when it's a Bayesian statistic, always the data has a strong enough of evidence, then the data is going to overrule the prior effect. What we find in practice is for QTL studies really not very different when you use different priors effects. But for GWAS, it could make a difference. And we usually, let the program, like the algorithm estimate is always the best thing to do.
Thanks.
So for what fraction of GWAS loci do you end up with, very small set, credible set at the end of running SUSY, for example? Do you have any estimates on that? And also, fraction of loci is the ELD pattern so tight that no amount of fine mapping can essentially resolve it.
So for the size of the credible set, it's always a power issue. For when you have a large sample size, okay, so when you have infinite sample size, as long as the two SNPs are not 100% correlated, in which case you can't do anything. But even if they're 99% correlated, you have a large sample size, you can pretty much distinguish them from each other quite decently. So then the question is, in GWAS, do you have enough sample to make the distinction? On that end, we do have some successful story with UK Biobank. I think with that large of a sample size, and also UK Biobank, you have the full genotype. You can pretty do a sufficient statistical version. So I don't have a table for it, but on average, the samples, the credible sites are pretty small. Sorry, I didn't hear your second part of the question.
No, I think that answers it. Thanks.
Okay, good.
I think in the interest of time, maybe we should move on to the next speaker. But thanks so much, Gao, for your talk. Omer Weissbrod is the next speaker. Omer, can you?
Yeah.
Okay,
Hi.
great. Yeah, so Omer is currently VP of Comp Bio and AI at 11 Therapeutics, and he was previously a postdoc with Alchemist Labs, and he has done a lot of work in integrating functional annotations with both fine mapping and polygenic risk scores, and that is very interesting from the point of view of IGVF as well, since we are all interested in functional annotations. So Omer, can you start?
Thanks. Sure. There you go. Okay, can you see my screen?
Yes.
Yep.
Cool. All right, cool. So thanks a lot for having me here. It's a real honor. So I'm Omer, and I will be speaking about leveraging fine mapping and multi-population training data to improve cross-population polygenic risk scores. And this is joint work with Masa Kanai and Wenbo Shi. And I did that while being a postdoc at Harvard with Alice Price, and it was published this year. So yeah, let's jump right in. So I'm going to start with this slide, this figure, which is pretty well known, I think, at this point. And what this figure shows is that polygenic risk scores lose a lot of accuracy in non-European target populations. And so this was published in a paper in 2019 by Alicia Martin et al. And what this figure shows is that when we train prediction models on European people, and then we apply the predictions to people with non-European ancestry, we get a big drop in the accuracy. And you can see here that when we train prediction models on European, as we go from left to right, you can see that we lose more and more accuracy because we get to populations that are more and more genetically divergent from Europeans. And when we get to the right, to people with an African ancestry, you can see that we only get a quarter of the accuracy that we got when we apply the predictions to Europeans. And this is, of course, a big problem because it means that we are expected to, it's expected to lead to very big healthcare disparities. And of course, this is a huge problem that we as a society have to fix and as a scientific community. And so this work is about our attempt to fix or to mitigate this problem. All right. So I'm going to speak about the introduction, why does this happen, and then our methods to try to address this, and then I will show our results. All right, so let's start with an introduction. So why does this happen? Why do polygenic risk scores lose accuracy across different populations? And broadly speaking, there are two main reasons for that. So it's LD differences and allele frequency differences. These are not the only ones, but these are the main ones. So let's speak a bit about each one. So LD differences, of course, exist between populations. Different populations have different LD structures between SNPs, and LD differences can lead to big loss in accuracy when we use non-causal SNPs to predict. So here is an example. So let's say that we have two populations, a European and a non-European population. And let's say that we have these two SNPs, SNP1 and SNP2, and we have a trait that we want to predict. And let's say that we are using SNP1 to predict the trait. And SNP1, as you can see here, it's in LD with SNP2 in the Europeans, but not in the non-Europeans. So what this means is that we can actually use SNP1 to predict the trait in Europeans, even though it's non-causal, because the effect is mediated through SNP2 and they're in LD. So we can get some predictive power by using SNP1. So what would happen in this case is that if we train a PRS model on the European population, and then we tried to apply these predictions on the non-European population, we would lose accuracy because SNP1 suddenly becomes non-predictive, right? Because it's not in LD with the actual SNP that we actually care about. So this is the first and I think the most major problem when we tried to apply these PRS models to different populations. And the second problem is due to allele frequency differences. And this happens even when we use causal SNPs to predict. So let's now say that we actually use SNP2 to predict. So SNP2 is causal, so all is good. But now let's assume that SNP2 is rare or maybe even absent in Europeans, but it's common in non-Europeans. So in this case, what would happen is that when we train a PRS model using a European population, European training set, so the PRS model just wouldn't be able to infer that SNP2 is actually relevant for the trait because it just can't see it, right, if it's absent. And then what would happen is that we won't be able to use it when we apply the predictions to the other population, even though in principle we could have used it. All right, so these are the two main causes of this loss of accuracy in cross-population PRS. These are not the only ones, but I think it mostly boils down to these two. And this realization already suggests a way how to fix that. So let's discuss how to fix this by addressing each of these causes. So what can we do about the LD-based problem? It's very straightforward in principle. We just need to use the causal SNPs instead of the non-causal SNPs to predict, right? Because then we wouldn't have this problem to begin with. And this is very intimately related to fine mapping, which Gao just described. So we're going to exploit this relationship. And then in order to solve the second issue, the issue of the allele frequency differences, in this case, we actually can't just use the European population. There's nothing we can do about it, right? Because if SNP2 is actually absent in Europeans, so no statistical tricks would help us get the information if the data is just not there, right? And so to address this problem, we really need to combine data from multiple populations. And so what we're going to do is basically combine these two approaches together. All right. So yeah, let's jump into the methods. So what we're actually going to do. Before jumping into the methods, I'm going to try to convince you that fine mapping is closely related to PRS applied to all SNPs. They're essentially the same thing. So this is not a math-heavy talk, but this is the only math slide in this talk. So all right, so let's assume that we have some trait Y that we want to predict, maybe some disease trait, or I don't know, blood pressure or anything. So we measured this trait for a bunch of individuals. So we have this vector Y, and now we assume, the most straightforward basic linear model. So we assume that Y is given by a linear combination of SNPs, and each SNP has an effect. So what this means is that we have this matrix X of SNPs multiplied by this vector beta of the SNP effects, plus some vector epsilon, which captures environment and noise and basically everything else. All right, so this is as straightforward as it gets. This is our model. And now let's try to think what we actually do when we do fine mapping. And this is not exactly the way that Gao described it, but I think it's essentially the same thing. What we do is we simply try to estimate the effect sizes for all SNPs jointly. So we have this vector beta, and ideally we just want to estimate this vector beta, all the entries in this vector beta, as accurately as possible. And that is it. If we could do that in principle, so beta I, its entry beta I would represent the causal effect of SNP I, and we would be done, right? And I should just add the caveat that when I say the word causal here, I mean it in a pretty loose sense. This is not causality in the Judah Pearl terminology. This is causality in the sense that the effect is not mediated by LD. It's a direct effect. So this is what we would want to do in principle, and this is what fine mapping does. And now what happens when we train a PRS model? So what we do in practice usually is first we select just a subset of the columns in this matrix X, and then we estimate beta for only the entries corresponding to the columns that we selected. So in this example, I selected these 3 columns, and then I try to estimate only 3 entries in the vector beta, and that's what people do in practice. And this is fine. It's not wrong per se, but what it means is that now its beta I represents a combination of 2 things. It's the direct causal effect, but it's also what we call the tagging effect, because this SNP tags the effects of all the SNPs that we omitted that are in LD with that SNP, all right? And this is what people do in practice. I don't think there is any, strong statistical justification. It just makes our life so much easier, and we can't really, jointly estimate a vector of tens of millions of SNPs. It's really infeasible. And what I hope that I convinced you is that fine mapping and PRS are at a conceptual level exactly the same thing. The only difference is whether we look at all SNPs jointly in some locus or only part of the SNPs. This is basically the only difference in a conceptual level. All right, so we're going to exploit this.
Omer, quick question. So why do people only look at a few SNPs for PRS instead of why not look at all like fine mapping?
So are you asking why people choose a subset of the columns? Because it would be too difficult, both computationally, and also there would be lots of statistical issues if you try to estimate like 10 million variables jointly. So yeah, if we had, a big supercomputer and infinite data, so it would be feasible. But in practice, there are many issues involved. I can already jump ahead and tell you what we did. We just ran lots of fine mapping on many different loci. So I forgot the number of loci already because it's been a long time, but thousands of loci, and we just ran fine mapping thousands and thousands of times. So we didn't really do it jointly for millions of SNPs. We took only several tens of thousands of SNPs at a time, and we used many different overlapping windows.
I see. Okay.
Sorry, can I follow up on that? Is the question, is the concern the computational speed at which you'd estimate all those 10 million variants, or is it that you don't have enough data to accurately estimate all of it?
That's a good question. I would argue that it's mostly the first option. So I don't think that not having enough data is a problem, because we regularize anyway, and we just need to regularize very heavily. But I think that the computational problem is the main issue here.
I guess I'm a little bit confused by that, because trying to estimate, particularly doing lasso on massive amounts of variables, is a long-standing problem in statistics, and there are a lot of methods to do that quickly.
I don't think that's a good question. I don't think that anyone ever applied lasso to the scales that we're speaking about, which is like 20 million variables with hundreds of thousands of rows, hundreds of thousands of samples. I mean, this is a few orders of magnitude, I think, more than what anyone has ever applied lasso to. Having said that, there are some papers that apply lasso to subsets of SNPs and report good results, but definitely not to all of the SNPs in the genome, which is what you would need to do in order to get this causal effect.
Well, I think that people have definitely applied lasso to problems bigger than this. It's just I don't think they've done it on like a laptop.
Maybe I'm not aware of what people did. I still think that it would be a huge overkill, and maybe someone should do that. But again, this is I think that the main issue is computational here. I mean, I think that we would run into all kinds of issues that we need to solve, but I don't think that there are any, conceptual reasons not to do that. If we could just, if we could just spend the computational effort.
Okay, thank you.
All right, so hopefully I've convinced you that PRS and fine mapping are the same thing, and so we just did that. So, sometimes there's a problem that you just ask yourself, why did no one ever do that? So that's what I said, and I didn't have a good reason, so I just went ahead and did that. And so we came up with this method, and we called it polypred. And so it's a bit trickier than that. And what polypred does is essentially it combines two things, a standard PRS and what we call a fine mapping PRS. And this allows us to account for LD differences. So let me walk you through what we have here. So let's say that we have a large sample, and usually, unfortunately, nowadays, that means that it's probably only European people, or almost only. And we're going to train two different PRS models. So first is what we call a standard PRS. So you can use your favorite state-of-the-art PRS method. We use mostly Bolt LMM in our paper. I will show you some results in a bit. And the second thing we did is we trained what we call a fine mapping PRS, which essentially does what I just described. So we just ran basically thousands of fine mapping runs across the genome with lots of overlapping regions. And again, we just combined all of these results together, and then we had basically two different PRS models, or two different estimates of the effect size for the SNPs in the genome. All right, so now we have this PRS model 1 and PRS model 2, and what do we do with them? So we need to combine them somehow, and we want to combine them in a way that is optimal for the target population that we actually care about, which might be people of non-European ancestry. And in order to do that, we need another training sample from the target cohort. So this could be people with a non-European ancestry. But in this case, it can be pretty small, because basically all we need is to combine two numbers, to find the optimal way to combine two numbers. So how to linearly combine PRS model 1 and PRS model 2. And so we can take a small training sample from the target cohort. And this is something that we can obtain because such small cohorts are available. And that is it. So we combine these two models together. And this is what we call the polypred PRS model. And it's conceptually simple, but I'm going to show you that it's a very powerful approach. So, okay, so this is polypred. And so this accounts for LD differences, but we still didn't account for the allele frequency differences that I described in the beginning. And so in order to do that, we have polypred plus, which extends what we just did. So polypred plus, I said that we also need another data set, another cohort, right, of non-European people with a non-European ancestry. And so this is exactly what we did here. And so if you can see the difference between this slide and the previous one, the only difference is that we added more stuff on the right. So the left is exactly the same, and we added some stuff on the right. So now we have another cohort of ideally non-European sample. Again, it should be ideally pretty large, otherwise the gains will not be very impressive. And now we're going to train a standard PRS model on this cohort on the right, and we have PRS model 3. And now we're doing basically the same thing. We're just combining these three models together, again, using a small training sample from the target cohort to find what we call the mixing weight. We just need to estimate three different numbers, and that is it. And in this way, again, we get very substantial improvements over the state of the art. So these are our methods, and feel free to ask me questions, because if not, I'll jump straight into the results.
This is very interesting. I'm wondering if, is it necessary to have labels in the test domain? Do you know what I mean? Like, is it enough to just know the LD matrix in the test domain and to make choices in the training scenario where you avoid these overfitting options, right? I mean, I guess it's not overfitting, it's like domain overfitting, right? Like in the training domain.
Right.
Or do you really need the test labels?
Yeah, so that's actually something that we looked into in our paper, and we actually just tried to take, fixed fixed combinations, for example, 50-50 or 25-75. And actually taking something like 50-50 is almost as good as what we get with the optimal combination. But the optimal combination is kind of like an insurance policy, because that means that you cannot lose, because you cannot be worse than at least the best of these two models, right? So I think about this as like an insurance policy. But yeah, you can just take some fixed combination like 50-50, and it will probably work in most cases almost as well.
I see. Okay, cool.
Yeah, I should mention, I mean, these are technical details, but we apply non-negative least squares to estimate these values, because this means that mathematically we are guaranteed that we cannot do worse than the best of these two models. We can always do at least as worse as the best of these two models, as bad as the best. Hope it's clear. Yeah, and one thing that I
have
want
a
to.
question.
Yeah, sure.
Sorry, I have two questions. So like, for your large non-European sample, why didn't you generate fine mapping PRS for that one?
Yeah.
And then my second question is, how your target cohort, like the small training sample, is it like what it consists of, or like how does that affect the optimization? Yeah.
All right. Let's start with the first question. Why only a standard PRS here and not also a fine mapping PRS? That's a great question. We could have applied a fine mapping PRS. It would be, a major effort, because this was a major effort, so it would be another major effort. And we hypothesized that the gains would be negligible. And the reason is that this other sample should ideally be from a population that is pretty close to the target cohort. And the closer the training sample to the target cohort, the less it matters if we do fine mapping or standard PRS, because the LD patterns are the same, right? The main advantage of the fine mapping is when the LD patterns are very different between the training and the test sets. So hopefully, if this training set is similar to this test set, so the fine mapping wouldn't give us that much advantage. Yeah, but we definitely could do that. There's no hard reason why not to do that, except that it's very complex and it would take a lot of time. So that was the first question. And the second question, can you repeat that, please?
Oh, yeah. I think you touched on it. Like, what the target cohort consists of, like which type of population or mix of populations, how does that affect the performance of your model?
Yeah, so, there's no hard definition. So what you would ideally want to do is to define a cohort of, a data set. So I think about it as a data set. So a data set into which you want to predict. And this data set, on top of the fact that these people probably have some, specific mixture of ancestries, there might be all kinds of, I don't know, artifacts or confounding factors or whatever that is specific to this data set. And this linear combination of these two or three PRS models could just be ideally fine-tuned to the specific properties of this specific data set. So I don't think that I can say anything more specific than that, but I will show you results in a few slides, and I hope this would make things clearer. Okay. So before jumping into the results, I just have a few slides about the functional annotations, because I know that this is something that people here care about. We actually spent a lot of time on how to use these functional annotations to help us in the fine mapping effort. But this was the subject actually of a previous paper to that one. So I will touch on that pretty briefly. So, when we do fine mapping, or when we ask ourselves what makes a SNP relevant, so we have two sources of information, right? So the first source is the GWAS, right? So we have GWAS data, and when we ask ourselves which SNPs are most relevant for some trait, so we look for the peaks in the GWAS, in the Manhattan plots, right? And these SNPs are the most relevant ones. And the second source of information is what we call functional annotations. So here is an example. So let's say that we have these traits of the genome, and let's say that for each SNP we have some annotation. So let's say that we have these three annotations: coding, intron, and conserved. And we can already say, without looking at the data, that these 3 SNPs are probably the most relevant ones because they're coding and they're conserved and they're not in an intron, right? And ideally, what we would want to do is to combine these 2 sources of information. So the function annotations and the GWAS data, and this fits very nicely into a Bayesian framework where the function annotations inform the prior distribution, and then we update the, the distribution, the posterior based on the GWAS data. And I'm not going to jump into the details. This was the subject of the paper that I wrote before that one. But I will say that we use lots of functional annotations coming from all these huge consortia that we have. So ENCODE and Roadmap and GTEx. And just to give you a taste of the types of annotations that we used in practice. So we have genomic regions, stuff like coding, promoter, and so on, and chromatin state, histone marks, conserved regions, enhancers, math beans, stuff related to the LD patterns, whether SNPs are cis EQTLs or not, and many, many others. You can find all of the details in the paper. And this is really a huge, huge collection of annotations that encompass many of the things that we actually know about genomes. And this gave us a big boost in our fine mapping power. Again, I'm not going to go into the details because this is the subject of a different paper, but just to give you kind of an intuitive sense of what it does. So we use functional annotations to improve fine mapping. And here is an example. I guess that after Gao's talk, you don't need that really. But let's say that we have, a bunch of SNPs here, and these are the significant, the p-values or whatever. And we know that probably one of these 3 SNPs in the middle is causal, right? Because they are strongly significant, but we don't know which one. But now let's say that we know their annotations, and we know that this SNP in the middle is coding and conserved, and the other ones aren't. So this gives us pretty strong confidence that this SNP is actually the causal one. So when we estimate the causal probability, we would be able to prioritize this SNP in the middle over the other ones based on these annotations, right? And so we use that, and we had a pretty complex setup of a Bayesian based framework using stratified eddy score regression and SUSY. We use SUSY a lot. Thanks, Gao, for that. And I'm not going into all of the details. And instead, I will jump straight into the results. All right, let's jump straight into them. So here are our main results. And the main result is that when we do that, when we use our method polypred, we get a significant improvement in PRS accuracy in the UK Biobank. And the bottom line, the punchline is that we get a 32% improvement in people of African ancestry versus Bolt LMM, which is, according to our tests, the state-of-the-art method. So let me walk you through what you can see here. So what we did is we took 7 independent complex traits. So this included traits like height and BMI and blood measurements and glucose levels and bone mineral density, and a bunch of traits that are really, really independent from each other so that we don't double dip. All right. And our training data was British people in the UK Biobank. So we took 325,000 people in the UK Biobank with British ancestry. So this was the average size of the training set. Okay, and we trained 4 different methods on this training set. And you can see the methods here in the colors. So we have polypred, which is our own method. And we have Bolt LMM, which not many people know, but it can also be used as a PRS model. And it's the best PRS model there is, except ours. And there is S-base-R, which is pretty similar to Bolt LMM, but it uses summary statistics, and that makes it a bit less powerful. And there is what we call P plus T, and P plus T is short for pruning plus thresholding. And you can already see here at the bottom that it does really badly. This is a pretty bad method, but still people use it all the time because it's so easy to use. It just means that you take the GWAS results, you just LD prune the SNPs, and you just take the marginal effect sizes. And yeah, it does really badly. Please don't do that. But actually, most of the papers, last time I looked, still use this method. Okay, so we have these 4 methods. We trained each of them using our training data of the British people. And then we applied these predictions to people in the UK Biobank that are not classified as British. So we had 4 broad population groups. So we had non-British Europeans and South Asians and East Asians and Africans. Okay, so these are our 4 broad population groups. And the quantity that we report, the y-axis, is what we call the European normalized relative R-square. And what this means is that we take the R-square and we normalize that according to how well we do in this orange bar here, basically. So how well does Bolt LMM do in non-British Europeans? Because we decided that this is going to be our baseline that we compare ourselves to. So you can see that this orange bar here, by definition, gets an average score of 1, right? Because this is the definition, okay? And then we compare ourselves according to that. And that allows us to not be, sensitive to the fact that different traits have different heritabilities and so on. And all right, so you can see the results, and you can see that polypred is always, the best. Surprise, surprise. It's actually not surprising because polypred uses Bolt LMM as one of its 2 constituent methods. So by definition, it has to be better. It cannot be worse. And you can see that the difference is statistically significant in all cases, except here in East Asians. And that's because the East Asian cohort was pretty small. It was less than 1,000 people. But when you look at the relative improvement, actually, the relative improvement, it increases when we go to the right, and it's actually largest in Africans. So as you can see in the title, for people of African ancestry, we got a 32% improvement over the state of the art, over Bolt LMM. This is what you can see here. So this is very nice in relative terms, but of course there's also a negative side, and the negative side is that you can see that in absolute terms we still do really, really badly for predictions of people of African ancestry compared to Europeans. So yeah, this is an important step in the right direction, but we still have a lot of work to do in order to address these gaps. Definitely. All right. So 2 more slides of results. So what we actually care about is whether we can predict well to other data sets, not to the UK Biobank, because there is some sort of confounding going on probably. And so we took these trained models and then we applied them to 2 other data sets, to Biobank Japan and to a cohort from Uganda called Uganda APCDR. And I'm showing you here only the 2 interesting methods, which is polypred and Bolt. And we can see that we get similar relative improvements. So again, polypred gets around 13% improvement over Bolt LMM in Biobank Japan. And it was pretty similar. I mean, it's not written. I think it was like 10 or 11% in Uganda APCDR. So the relative improvements are nice and encouraging, but again, there is a negative side. And the negative side is that if you look at the y-axis, you can see that it only goes up to 0.3, meaning that in absolute terms, we are doing much worse across the board when we predict to different cohorts. And this is a general problem. This is not something specific to our method. In the UK Biobank, people always get much better accuracies than when predicting outside of the UK Biobank. And in the paper, we looked into that a lot and we tried to come up with an explanation. We're not sure that we nailed it, but we think that it's probably because everything is just more heritable in the UK Biobank for reasons that you can debate. But when you estimate heritability in the UK Biobank, all of the traits have a much larger heritability in the UK Biobank compared to other cohorts. Don't ask me why. So you can look at the paper for more details. So this is the the glass half empty side. But again, in terms of relative improvement, we got very nice and consistent relative improvements versus the state of the art. And last result. So what I showed you up until now is the method called polypred. And I said that polypred only accounts for LD differences, but in order to account for other sources of differences, we need to have another cohort. And so we have this method called polypred plus that uses multiple cohorts. And what we did here is we used polypred plus and we had 2 training data sets. So we had the same training set as before of UK Biobank British. And on top of that, we added another training data set of Biobank Japan. So we had 124,000 people from Biobank Japan as a second data set. And we trained PRS models on each of these training data sets separately. And then we combined them and we applied the predictions to people of East Asian ancestry in the UK Biobank because they are the most similar to people from Biobank Japan in terms of their genetics. And sorry, the colors are mixed here, sorry for that. But now Polypred Plus is blue, and you can see that Polypred Plus gets a very impressive improvement, relative improvement versus Polypred itself, and of course also over Bolt LMM. So actually in this case, the improvement of Polypred of orange versus green is not significant, but the improvement of blue versus green, so Polypred Plus versus Bolt LMM, is statistically significant, even though it's a pretty small cohort of only 900 people. So yeah, Polypred Plus does a very nice job in improving over Polypred itself. And yeah, so I'm going to wrap it up. So we came up with two methods called Polypred and Polypred Plus, and Polypred leverages fine mapping to improve cross-population PRS, and that gives it a 32% improvement versus Bolt LMM in UK Biobank people of African ancestry, and 11% over Bolt LMM in people of East Asian ancestry. And on top of that, we also have Polypred Plus, which on top of the fine mapping also leverages multi-population training data, and that gives it a 24% improvement versus Bolt LMM in people of East Asian ancestry in the UK Biobank. And yeah, that is it. And I want to thank many wonderful people, especially Wenbo Shi and Masa Kanai, who are joint co-first authors of this paper, and Alkit Price, my postdoc mentor, and many other great people, and the UK Biobank for being such an amazing resource. And thank you.
Thank you so much, Omer. I think in the interest of time, maybe we should move to the next speaker, but you can ask Omer any questions on the chat. I think we can answer this there.
Sure. How do I stop sharing? Yeah.
Thanks so much, Omer. This is really interesting and fascinating. So I think it also plays well into the next talk. So I'll introduce Yun. If you wanted to, you could maybe, Yun, you could already try to show slides. So
my screen, okay?
Yeah, so I'll do a quick introduction. So we have Dr. Yun Lee from UNC Genetics Department and Biostatistics Department. I'm sure, probably everyone here has already interacted with Yun through the consortium, but just to mention, she's very active in numerous consortia, including TopMed, and has probably really useful insights and tools from that consortium that play well with IGVF. And also she recently was awarded a U01 with a number of other researchers from NHGRI to improve utility of polygenic risk scores in diverse populations. So thanks so much for sharing with us today.
Thanks a lot for the invitation. Thanks so much for organizing the workshop. It's great. Really enjoyed all the discussions and the presentations. So, like my talk, I think I'm Omer, and I feel like people don't need more methods for the fourth talk in the afternoon. So I will mostly focus on findings and some of the figures. But the theme of my talk today is focusing on the power of diversity in genetic studies. So basically, here's a brief outline. I will first, talk about why diversity matter. It's definitely, not only for political reasons. There's really great scientific reasons for us to leverage diversity and try our best, to promote diversity in genetic research. And then I will actually talk about one resource we developed the most recently. So I think one theme that has been come up consistently from all the three previous speakers is linkage this equilibrium here, LD, the importance of LD, and LD score regression, as the name suggests. So LD basically underlies almost all the relevant method we talked about. So I'm going to introduce a resource that we developed recently using the whole genome sequencing data from the TopMed consortium. We name it TopLD, and then at the very end, I will just use one slide to introduce you again the diversity focus group. I'm sure a lot of you have heard about that. I will only remind you with one slide, but I really want to bring people to the attention to that focus group and welcome your feedback and your participation. So here we go. So as we know, genetic studies, although GWAS have been hugely successful, but we know the vast majority of genetic association studies have been focusing on European ancestry individuals. So here's a figure pie chart from about 3 years ago, Nature Reviews Genetics. As you can see, only 22% of the GWAS participants are from non-European ancestry individuals. Well, if you look at the findings, 46% of the GWAS loci identified are from the non-European ancestry, findings. So, really, they're not only, very important in terms of health disparity perspective, but they are actually truly powerful in our, genetic mapping and genetic studies. Omer have already showed this figure, so I'm showing this again. This is another excellent figure to show, how underrepresentation of non-European ancestry individuals have lead to health disparity, reflected by polygenic risk score. And then you can see African ancestry individuals compared to European ancestry individuals have much lower predictability using the PRS constructed using the current published GWAS results. We know a lot of drugs, at least certain drugs, are less effective, even unsafe in some populations and some diverse ancestral groups because of the genetic differences. So it's not only necessary, but absolutely urgent actually for us to take this into serious consideration to avoid a further exacerbation of health disparities. And then I will talk about, the power of diversity in 3, aspects. There are actually many good reasons, again, like I said, scientific reasons as well, to expand the diversity and to promote the diversity. I will just focus on 3 different aspects. First is the detection of novel genetic loci from genetic association studies. I'll give 2 examples here, and then I will give 1 example in terms of identification of effects that are differential across ancestral groups. And finally, we'll talk about the power of diversity in terms of pinpointing causal variants through fine mapping methods. So first, I want to introduce this study that we published about 2 years ago in Cell, which is a multi-ethnic study for blood cell traits as part of the blood cell consortium led by Guillaume Latre, who is also IGVF investigator. So in this study, we total studied through about 750,000 participants, as you see here. Still, over 500,000 from European ancestry, but you see, quite a sizable from East Asian, 150,000 individuals and 15,000 African ancestry. So covering diverse ancestral groups. So basically, this is what we found, summarized in terms of which, whether it's reflected in multi-ethnic or trans-ethnic, a meta-analysis or captured by each or the combination of the different ancestral groups. So as you can imagine, the vast majority are actually driven by European ancestry individuals. But I want to bring your attention, to those colored arrows here as the loci that are, identified primarily driven by non-European ancestry individuals, but not in European ancestry individuals. For example, those green arrows are basically loci that are driven by, East Asian ancestry, and some of those are even not significant when we bring in the other ancestry. So the signal, in some sense, it's washed out, and none of those that are highlighted have any signal from the over 500,000 European ancestry individuals. So basically, this showcases the power of using non-European ancestry individuals, even with a much smaller sample size. We're discovering novel loci that were missed by European ancestry individuals. So again, consistent with the pie chart I showed you earlier, maybe it's only a small percentage of the GWAS samples, but they are contributing almost 50% of the GWAS loci reported in the literature. So here's a second example I'm going to show in terms of the identification of the novel variants. So this work is led by a graduate student, Chuan Sun, and a colleague, Laura Raffield. So we have been working very closely. So in this study, we performed GWAS for serum and urine biomarkers, about 31 of those biomarkers in the UK Biobank non-European ancestry individuals. So as people know, in UK Biobank, about 500,000 individuals, the vast majority are European ancestry individuals. So there's over 400,000 European ancestry individuals, but there are still, about 10,000 African ancestry individuals, 10,000 South Asian ancestry individuals, and about, slightly below 3,000 East Asian ancestry individuals. So basically, we're just doing GWAS using those much smaller sample size, close to 10,000, 3,000 to close to 10,000 individuals. So I want to show, there are several novel findings. We have, I can't remember the exact number, I think 30 or some novel findings that were missed by the much larger, European ancestry and by other studies reported in GWAS catalog. But here I'm going to show you, one example for hemoglobin A1C in South Asian ancestry participants in the UK Biobank samples. And here's a mirror Manhattan plot. On the bottom, it's just the plain unconditional GWAS results. So for people who are not super familiar with this, the y-axis is negative log-term p-value. So the higher you see, the more statistical significance they correspond to. And the red dashed lines mark the genome-wide significance, 5 times 10 to the negative 8. This is unconditional. Basically, for each GWAS variant, we figure out their association with the hemoglobin A1C levels. And the top panel will basically conditional all the variants reported by GWAS catalog, as well as all the variants discovered by European ancestry individuals in UK Biobank. As you can see, two loci remain genome-wide significant even after conditional known variants and variants reported or discovered in European ancestry individuals. Again, European ancestry individuals in UK Biobank, it's over 400,000 individuals. So it's at least 40 times more than the non-European ancestry individuals like we evaluated. And it's 100 times more than the East Asian actually. So just to look at those two loci, this one on chromosome X, and I also want to mention in a lot of genetic association analysis, you don't see chromosome X, you only see the autosomes. So this is another a gap in genetic studies. So here, as you can see, we have a very strong signal even after conditional analysis. And again, in South Asian, we have a slightly less than 10,000 individuals. So it's not a huge sample size. But as you can see, the p-value is super significant, less than 10 to the negative 30. So this is the G6PD gene, as people might be familiar with, and the index variant is a coding variant, and not reported to be associated with hemoglobin A1C, since it's about conditional analysis. But other coding variants in G6PD have been previously reported to be associated with the hemoglobin A1C. So the signal is very likely to be true, and pointing out to G6PD is actually quite plausible because it has been known hemoglobin A1C measurements is affected through erythrocytic means, so through red blood cell. So yes, we know hemoglobin A1C, as the name suggests, is hemoglobin. And not only this finding is significant, we discover a novel variant, but this also has very important implications, consistent with the literature, this actually brings or adds to the evidence some concerns over, using hemoglobin A1C to measure glycemic control. And in, certain ancestral groups, including, African ancestry, and here showing South Asian ancestry, when there's, variants affecting, in those red blood cell genes, that can affect, like how accurate hemoglobin C actually measures the glycemic control. So this actually brings, like it has important implications in terms of clinically how we should use hemoglobin C and whether we should adjust out certain genetic components to better reflect the physiologically relevant dynamics relevant for clinical decisions. And the second locus right here on chromosome 16 is mapped to the PIZO1 gene. It's a non-coding variant that is has a relatively high conservation score. The CAD score is almost 10. As people know, PIZO1 gene encodes erythrocyte membrane protein, and other variants in the gene have been reported to influence, like red blood cell dehydration and associated with decreased malaria infection risk. Again, it's likely affecting hemoglobin A1C levels through erythrocytic means. So again, like I mentioned multiple times, the South Asian ancestry participants in the UK Biobank is only slightly less than 10,000 individuals, much smaller than the UK Biobank European ancestry individuals. But you can see, we still discover like novel variants that also can have quite clinical significance potentially. So with that, like kind of that's the first aspect I want to talk in terms of power of diversity. The second aspect is regarding, like diversity can basically reveal potential differential effects across the diverse groups. And here I'm showing, several different two papers published recently, where basically reported, and we all know like ApoE variants, particularly the like E2 and E4 variants exert higher risk of Alzheimer's disease for individuals carrying those alleles. However, like we didn't know how much those variants, those alleles have differential effect across ancestral groups. And those two recent papers, basically by analyzing individuals of African American and Hispanic Latino individuals, show that show consistent evidence like those risk alleles when inherited from African background tend to exert less severe effects than when they inherited from the European ancestry background. I mean, what's the mechanism? We don't know yet, but like I think that's the power of the diverse samples. They basically pinpoint to us certain genes or genetic variants that exert differential effects across different groups, which like I think would be great for us within IGVF to follow up. So that's the only example I'm going to give for this second aspect. And the last aspect, I think many people have talked about fine mapping. But like we haven't really emphasized using diverse samples to help with fine mapping. So I mentioned earlier the first like a powerful aspect is to identify novel variants. But like diversity can also help pinpoint the most likely causal variants, and also for the same reason can help with building more transferable polygenic risk scores. So here's one example where like basically the association around the DMT gene for like intronic variant, so the index variant or the variant with the most significant p-value with LDL cholesterol is this one shown here, the purple diamond. So Gao nicely already showed this so-called locus zoom plot, so I won't need to explain what that means. And here's the locus zoom plot in European ancestry individuals. So similar to the plot Gao showed, you see like a bunch of variants that's in high LD with the index variant also shows a highly significant p-value. So we're really talking about p-value between 10 to the negative 20 and 10 to the negative 25. So with the association signals alone, it's hard to figure out which is the causal variants versus which are just simple LD surrogates or LD tags. So in this study published in Nature from the Global Lipid Consortium, they basically leverage the samples from diverse ancestral groups. So besides European ancestry individuals, they also have admixed African American individuals. So as you can see here, the same variant is also most significant. But you can see here it didn't pass genome-wide significance, the magic 5 times to the negative 8. So as you can imagine, when we pull the samples together, not only this variant is genome-wide significant, but it actually distinguishes itself from the rest of the markers, basically effectively helping distinguish the more likely causal variants from its LD tags by using differential LD patterns across the 2 ancestral groups. And it also shows better, a co-localization with the EQTL for the DMTN gene. So it's not only helping, identifying the most likely functional variants, but also help pinpoint the targeted genes, which may not be, and in this case, it is, the closest gene, but a lot of the time the targeted gene actually may not be the closest gene. So helping with the co-localization with the QTL evidence is also, a big benefit of fine mapping using diverse samples. And as I mentioned earlier, the power is largely attributable to differential LD patterns. So here I'm showing, actually in the paper, show, LD, and on the top is from the African and samples, and the bottom European samples. It's not surprisingly, it's very typical that you see, more extensive LD in European ancestry individuals than in African ancestry individuals. So, LD actually is a, double-sided sword. When, in populations where you have high LD, it helps you to identify the region carrying the causal variants using, by directly examining a much fewer number of markers. But at the same time, when it comes to fine mapping, it hurts you because you just can't tell, the causal variant from its LD tags. While using differential LD patterns across different diverse groups, it will help, pinpoint the most likely causal variant. So here's another example from our multi-ancestry GWAS study for type 2 diabetes. And similarly, using, multi-ancestry fine mapping, mainly here, European and East Asian samples, we were able to pinpoint the most likely causal variant to only 3 variants. And as you can see from the bottom here, the 99% confidence sets, and in two functional regions. And the one on the right basically overlaps with the promoter region of the PROX1 gene, and the 2 on the right, both fall into the same enhancer region. And they are, enhancer or promoter in both pancreatic islet and liver. So highly relevant and are likely to be the functional variants. And again, from large number of variants showing genome-wide significance at the loci in both European and East Asian ancestry, the ability to pinpoint only to those 3 variants is largely thanks to differential LD patterns across the different ancestries. I'm not going into details, but you can see, how the same, plots colored by LD in different ancestral groups differ, tremendously. For example, here, the largest LD surrogates for the index variant here are here in European ancestry, but East Asian here, while you don't really have any, a very strong LD tag in African ancestry individuals. So with that, I guess, like I will spend the last 5 minutes or so introducing the tool I mentioned earlier, which is the top LD. We name it top LD. So it's basically a tool to explore LD patterns using the NGLBI founded transomics of precision medicine, or TopMed whole genome sequencing data. Again, it's using TopMed whole genome sequencing data, and we focus on 4 continental ancestry groups. So I would like to highlight several aspects. First is like our top LD catalogs a much more complete set of variants, particularly for lower frequency variants. So shown here are for the 4 ancestry groups, the number of variants that's in top LD, which is a right bar here. Compared to haploreg. So in our data, we basically break down into common variants with minor allele frequency over 1% versus low frequency or rare variants with minor allele frequency less than 1%, and compared with haploreg, which is based on the 1,000 Genomes Project, total around 2,500 individuals, about 500 individuals from each of the continental groups. So as you can see, we have a much larger number of variants documented and with LD provided, in particular the true for European ancestry individuals and African ancestry, for African continental group. For European, we have a total 13,000 individuals. For African, we have 1,300 individuals, again, compared to 1,000 genomes, it's about 500 each. But you also see gains in the 2 Asian populations as well. So that's the first big highlight. The other highlights include that we included the chromosome X. As I mentioned earlier, many studies actually ignore the chromosome X, which, as we see from the hemoglobin A1C example, chromosome X contains actually very important genes. And we also included the structural variants. So shown here, for example, for each of the ancestral groups, we included somewhere between 17,000 to 79,000 structural variants. And we evaluated both the LD among themselves and also LD between structural variants and SNPs, single nucleotide variants. We sort of mentioned, Gao, talked Susie and fine mapping methods in general. So here we basically evaluated how, information provided by top LD can help with the summary statistics based on fine mapping. And in particular, we basically analyzed this particular GGT1 locus that we have been found previously. So it's from Chuan's paper I showed you earlier, where we performed the GWAS for biomarkers in UK Biobank. So in particular, we found variants at the GGT1 locus associated with gamma-glutamyl transferase. So not surprising, as you can see from name, this is a gene encode for this protein. And we found actually strong signal in the African ancestry participants in UK Biobank. So, Gao already told you about, the caveat regarding, conditional analysis. But, based on our individual level data, this is how we performed the sequential conditional analysis, where we're kind of treating as the working truth. Of course, one thing, we can do to confirm is basically to use, the multivariate model to basically confirm the percentage of variation explained in the outcome protein abundance. But here's just what we did. So basically using individual level data, we established basically those 7 variants that through unconditional analysis and the sequential conditional analysis that are associated with the GGT. Basically, it's we identified 7 distinct signals at this locus. And then we want to see, how much, does the LD from different reference panels, basically the choices from our top LD or previously from 1,000 genomes help with fine mapping based on summary statistics. So here's what we find. So we ran the fine map software. Again, this is just based on GWAS association results, not leveraging any functional annotations. So we use a fine map generate 95% credible set using the LD information either from the 1,000 genomes reference or from our top LD evidence. And actually, in both cases, the 95th credible set ended up to have exactly 5 variants. That's a coincidence. But the 5 variants have only one overlap, which is this variant here. This variant is actually the most significant variant as established from the working truth, as you can see here. This variant has any conditional p-value 10 to the negative 61. So if regarding the first, distinct signal at the locus, both reference, LD reference identified without any problem and then highly consistent. But then if you look at the other, variants identified from summary statistical based fine mapping using 1,000 G reference, it's that they are all partially reflecting the first distinct signal. You can see, they are not in an LD with any of the other 6 distinct signals, but rather they are in LD to various extent, from 0.3 to 0.6 with the first distinct signal, this RS40 variant here. In contrast, when we use top LD as reference, we were able to, recover 3 of the 7 distinct variants established from individual level genotype data. And then we were, if we allow LD tag, we actually, discover a 4th one where, although the credible set variant is not the variant itself, but it's in high LD with the LDR square over 0.83. So, basically using 1,000 genomes summary, will only it only capture 1 signal out of 7, and regardless of whether we use LD tags. And while using top LD, we can recover 3 signals out of 7 without LD tags. And if we allow LD tags, we're able to recover 4 of the signals. So as promised, we use the last minute just to bring your attention again to the focus group impact on diverse populations that Xinhua and I co-chair. So in this talk so far, it just focused on different ancestral populations, but in our focus group, we also consider different sexes. I think both the ancestral population and sexes would be the focus of this second year. But we can also broadly include socioeconomic groups, different animal strains. So with that, I'd like to thank many people in my lab who contributed to the work, and of course, thank Top Med for providing the data and prime the consortium, and of course, IGVF. And thank you very much for your attention.
Thank you so much, Yun. We have a couple minutes maybe for some questions. I have one to start. Is if you already knew some loci, associated loci for a GWAS, and you wanted to go through and maybe narrow the candidates, is there a technique to, or how would you, if you could prioritize how to balance sample size across populations, would it just all go to populations that have low LD?
Yeah, I think that's an excellent question. I don't know. The honest answer is I don't know. I think it really depends. Yes, low LD will give you fewer candidates to start with. But imagine the causal variant has lower allele frequency in the low LD population, and then you start with low power. So Yeah, and also that also depends on genetic architecture. This is, I think, pretty much all I talked about are kind of making the implicit assumption, like the causal variants are shared across the ancestry groups.
Right. Yeah.
May not be true. If that's, I mean, of course, I think it's largely true based on the a lot of empirical findings and also based on biology, right? So most of the causal variants are shared, but there are examples where in different populations, in particular when we get to the lower frequency rare variants, there are population specific variants that contribute to the disease etiology. So when that is not true, it's even harder to kind of balance between sample size and across the populations. So yeah, I don't have the good answer, but like my argument is like we should try our best to include the non-European ancestry individuals because they are severely understudied. So like for European ancestry, for a lot of traits, like unless they're really very small sample size, we should have already seen them. But like for a lot of other populations, so a lot of cases, like although it's a hard question, but in a lot of sense, it practically may not be the question because they just don't have many non-European ancestry individuals to select from. So yeah, that's hopefully that answers the question.
Yes, thank you. Perfect.
I have a question. Is that the same reason why, like in your analysis, you were able to recover 4 friends, 4 LD buddies, but you didn't recover all 7? So like you didn't recover the other 3. Are those possible reasons, or are there other additional reasons why the other 3 are missing?
Right, that's an excellent question. We haven't looked really deep into that. I suspect the lower effect size is another, like issue for the, for the, like signals that are lower down in the list. But yeah, I agree. I think what you talked about, so like the ancestry, more ancestry specific effects can also be in Asia. Here, I mean, like it's all in African ancestry individuals. But that said, like in UK Biobank, it's African ancestry individuals. While like when top LD, we focus on individuals with over 90% African ancestry. So actually even the global percent of African ancestry distribution differ between the target sample and the top LD. So that's actually a big challenge for LD and mixed populations. So I was about to ask Gao, but like I think he's gone. He mentioned all the caveats of using like in sample LD versus reference LD. But for mixed populations or multi-ancestry populations, even using in sample LD, I mean, does that really make any sense?
Any other questions for Ewan or for any of the other speakers? I think Gao is probably not here, but if there are any other questions.
Yeah, I actually have a question for Omer. I just wonder, when you used to, constructed the PRS, have you tried, are you trying in those like at mixed populations, and what are your findings? I kind of
I'm not sure I'm following. Can you please repeat?
Oh, at mixed populations, like how much like a fine mapping help with the construction?
Yeah, that's a great question. Well, I don't have a simple answer. I guess I have a complex answer. So the complex answer first says that, we never tested it directly, but we know that actually individuals in the UK Biobank of African ancestry are actually admixed at some level. So this is already a proof of concept that the method is applicable to individuals of admixed ancestry. We do think that for such individuals, you can come up with more, clever methods that directly exploit the admixture patterns in these people, because some SNPs are actually European. So some haplotypes are European and some haplotypes are not European, and you can actually exploit that. We didn't do anything like that. There are several methods out there that do exploit that. So we can probably do better than that.
Yeah, thank you.
Sure.
Any other questions? Okay, if not, so this is the end of day 1 of our workshop. There is going to be a day 2, which will be on November 17th. The schedule is already there, but there may be slight changes to the schedule, keeping in mind the camp working group as well. But thanks to all the speakers, thanks to everyone who joined in for this day 1 of the workshop, and the recording of this of today's talks will also be available at the same Google Drive location that we just shared. We'll also post it on maybe the camp channel or some of the other channels on Slack. Thank you. Thanks, everyone. Have a nice day.